Welcome back to So We Learn. Today we are going to study with another new topic called Stelar evolution. Stelar evolution. The stele is a Greek word meaning pillar or column, which is defined as the central vascular cylinder with or without the pith. Endodermis is the boundary between the cortex and stele. The central cylinder or core of the vascular tissue consists of xylem, phloem, pericycle, and the medullaries and pith is technically called the stele. The vascular system concept was put forward by Van Tegman and Dan Liot in 1886, who proposed the and developed the Stelar theory. The stele of the stem was connected with that of the leaf by vascular connections known as the leaf threads. In pteridophytes, the stele is differentiated into two major groups. One is known as the protostele and the other is known as the siphonostele. Protostele is the simplest and the primitive type. In this, the vascular cylinder is a concentric solid mass, and the central core of the xylem is surrounded by a layer of phloem and then by a layer of pericycle. The protostele exists in the following types. The first is known as the haplostele, which is the most primitive type of the stele. In this, the xylem forms a solid, smooth, and spherical central core, which is surrounded by continuous concentric layers of phloem, like in Lycopodium and Selaginella. The second type of the protostele is known as the actinostele. In this modification, there is a central xylem core which has radiating ribs, and phloem is not concentric but present between the radiating ribs of the xylem found in xylotem. The protoxylem is present at the tip of the radiating arms. Then the third modification of the protostele is known as the plectostele. Here the xylem gets broken into number of plate-like lobes which may or less lie parallel to one another, and the phloem alternates with the xylem plates. Example is Lycopodium volubile. Then the other type of the protostele is known as the mixed protostele. In this, the masses of xylem and phloem are uniformly distributed. Scattered groups of xylem embedded in the ground tissue of phloem, as in case of Lycopodium cernum. The protostele with mixed pith. In this, the center of is parenchyma cells associated along with the trachea cells. In example is lepidodendron. It may be derived from the transformation of the tracheids into parenchyma and may be first step in formation of the pith. Such type of protosteles is termed as mixed protosteles. The second type of the stele that is found in pteridophytes is known as the siphonostele. In this, the medullated protostele is called the siphonostele, which is characteristics of the class Filicophyta. It is the modification of the protostele. During the development, the siphonostele, the central core of the xylem, is replaced by parenchymatous cells, so the different pitch surrounds by xylem appears in the center. On the basis of the branches and leaf gaps, Jeffrey distinguished two types of siphonostele. One is known as cladosiphonic siphonostele, where the leaf gaps are not found, and the other is known as phyllosyphonic siphonostele, where leaf and branch gaps are present. According to the origin of the pith of siphonosteles, two theories were being proposed. One is known as interstellar origin of the pith, given given by given by Bower or given by Bower and Jeffrey. According to this view, pith has originated as a result of transformation of tracheal elements of the central xylem core into parenchyma. This pith is Intrastellar, this view was supported by Baudelaire in 1901, Wangani in 1908, and Bower in 1911, and is found in Osmunda regalis and Botrychium ternatum, which where tracheids are scattered throughout the pith. In extrastellar origin of the pith, which was supported by Jeffrey in 1902, 1910, and 1970, according to this, protostele has transformed into siphonostele due to the migration of the cortical cells into the stellar axis. Opening such as leaf and branch gaps possibly provided passage for invasion of parenchyma. This use derives support from amphiploic siphonostele that has two endodermal layers. One delimits the stele from the cortex and the other from the pith. Since endodermis is the structure peculiar to cortex, the pith is considered as cortical in origin. Jeffrey classified siphonostele again of two types on the basis of the position of the phloem. One is known as the ectoploic siphonostele and there is known as amphiploic siphonostele. In ectoploic siphonostele, the pith is surrounded by concentric xylem cylinder and next to xylem the concentric phloem cylinder. The phloem is restricted only in the external sides of the xylem. Example is osmunda and the pith is central in position. In amphiploic siphonostele, the pith is surrounded by vascular tissue. The concentric inner phloem cylinder surrounds the central pith. Next, the concentric xylem cylinder, which is immediately surrounded by other phloem cylinder. It means phloem is present on both sides of the xylem found in Marsilia. So these are the figures showing ectoploic siphonostele and amphiploic siphonostele, where in ectoploic you can see the pith surrounded by xylem and then by the phloem and pericycle and endodermis. While in amphiploic siphonostele you can see the phloem present on both outer and inner side and then follow in between is present the xylem and in center is the pith. The siphonostele modifies and forms the solenostele, where the siphonostele perforated the places or places to the origin of the leaf traces. Such condition is known as solenostele. They are again of two types. One is known as ectoploic solenostele, which has been derived from ectoploic siphonostele. Here there is a phloem is present only on the side of the xylem. In amphiploic siphonostele, this is derived from amphiploic siphonostele. So this is the phloem is present on both sides of the xylem. This is the solenostele and amphiploic solenostele. Then the other type is of modification of the siphonostele is known as the dictyostele. This is the advanced type found in Pteropsida, where the successive gaps may overlap each other. So Brebner in 1902 called the siphonostels with overlapping gaps as dictyostele. So in this case, the solenostels breaks into a network of separate vascular strands due to crowded leaf gas. In such cases, each separate vascular strand is known as meristele, and each meristele is protostelic type. Then the other is known as the eustele. According Brebner, there is one more modification of the siphonostele known as eustele. Here the stele is split into distinct collateral vascular bundles. So the vascular bundle systems of the ring of collateral or bicollateral vascular situated on the periphery of the pith. So collateral vascular bundles are those bundles in which the xylem and phloem are forms where they together and in between is present the pith, the cambium, and the cambium may, if not present, we call it as the closed collateral vascular bundles. In bicollateral, the xylem is surrounded on both sides by the phloem. In steels, in eustele, the interfascicular areas and the leaf gaps are not distinguished from each other very clearly. So this is an example of the eustele is equistem. Then the other modification of the siphonostele is known as atactostele, where the vascular strands are scattered. It occurs in monocotyledons. Brebner in 1902 coined the term atactostele, where atacto means without order, for vein arrangement seen in transverse view, which have been described later as scattered by Berg in 1997. Polycyclic stele is is a type of the stele in which the vascular tissues are present in the form of two or more concentric cylinders. Such a stele is called polycyclic stele. A typical polycyclic stele possesses two or more concentric rings of vascular tissues. This may be solenostele or dictyostele. Two concentric rings of vascular tissues are found in the pterygium equilibrium and three in Matonia pectinata. Polysteles, sometimes more than one stele, are present in the axis of some pteridophyte. Such a condition is called polystelic, and such steles shows polystelic condition. Certain species of Selaginella have polystelic condition. So this is the polystelic conditions found in Selaginella stem, where you will find more than two vascular bundles in the pith. So if you see the evolution of the stelar system, it is clear that the simplest type of the stele is the protostele, and from the fundamental type of the vascular plants in general and pteridophytes, all other types of the steles have been derived in the course of evolutionary specialization. Pteridophytes in the initial stage starts with a protostelic stem. When the shoot grows, accompanying with an increase in the size, there is internal differentiation in the stele, and the first step is appearance of parenchyma scattered in the xylem core, and finally a central parenchymatous medulla or pith is developed in the protostele, such type of the stele called as siphonostele. The method by modulation came about into protostele and form a siphonostele is a debate question and explained by two hypotheses called extrastellar and intrastellar origin of the pith. In simplest form, the siphonostele has no leaf gaps. Selaginella is known as cladosyphonic siphonostele, while in contrast with phyllosiphonic conditions with the leaf gaps. Siphonosteles which were perforated by scattered leaf gaps are known as solenosteles, and a siphonostel with more overlapping gaps are known as dictyosteles. The elaboration of the stellar organization in pteridophytes consists in the development of number of separate steles. Such a stele is known as polycyclostele. Another modification of cypnostele is the eustele, in which the vascular system consists of collateral vascular bundles. So this is the evolution of the steles, where you see from the protostele. In one section, they will develop into actostele followed by plectostele, while in another level from the protostele, amphiploic siphonostele develop. From the amphiploic, it will form the dictyostele, and from the dictyostele, it forms the atectostele. In another shape, we see from the protostele develops the ectoploic siphonostele. This ectoploic develop into eustele, and then from the eustele is being developed, and from the eustele develops again the atactostele. Thank you.